Our last speaker is Dr. Gedaly, who received his, Roberto Gedaly received his undergraduate and medical training at the Central University in Venezuela and Caracas. And then we normally think of someone as the triple threat who excels in education, research, and clinical excellence. But Dr. Gedaly is a triple threat in that he did 3 fellowships in the United States, all in transplantation. Dr. Gedaly, take it away.
Yes. So I would like to thank Dr. Nabnet for letting me come here this morning and talk about what we have been doing at the Transplant Center in the past couple of years. But I'm going to focus on a collaboration that we have ongoing with biomedical engineering. And I'm also gonna at the end show a slide of a potential collaboration in the future with biomedical engineering. So in the last couple of years, in our center, we have worked hard on basic science research and clinical science research, mostly on cancer, hepatocellular carcinoma drug development, where we actually hold 2 patents on more than 200 compounds with the biomedical, biochemistry department. These compounds have anti-cancer properties, but also we're claiming immunomodulatory effects. And most of our work in the last 7-8 years has been in immunology. I started working with Dr. Marti around 8 years ago when he was actually at the Department of Microbiology and Immunology. And most of our work has been on repurposing molecules for immunosuppression. But in the last 5 to 6 years, a significant amount of work on T cell biology and specifically on a subgroup of T cells that is called suppressive or regulatory T cells that are critical in allograft tolerance. So in clinical research and in our center, we have several multi-centric clinical trials, but we have 3 investigator-initiated trials, and I'm going to focus on the Tolerance Induction trial that is now ongoing in kidney transplant recipients and is funded by Novartis, FDA approved and IRB approved. So without the intent of giving a lecture of immunology, that would be too long to talk about these cells. These are the FOXP3 cells called regulatory T cells. This is actually one of the main markers of these cells. They usually come from the thymocytes in fetal life, and the pool of those cells are called natural Treg cells. They can also come from naive T cells and effector T cells. The effector T cells, that they have that plasticity that can become regulatory T cells, and the Treg cells actually have the same plasticity. They can become effector cells. They represent around 5 to 10% of the CD4 cells. So some of these work comes from the from a couple of publications in the literature where the ratio between effector T cells and regulatory T cells is what really induces tolerance in the allograft. So if you actually remember the drugs that we use in current clinical setting, most of these drugs target effector T cells. So Prograf, cyclosporin, Cellcept, mycophenolic acid, and steroids, and also thymoglobulin basically target T cells. One of the problems with these current immunosuppression regimens is that they're very toxic. We, in the last couple of years, and other groups in the United States and also around the world, have been working on trying to enhance levels of regulatory T cells to induce immunosuppression and actually induce tolerance. This is a 1-minute video that summarizes the trial, which is where we're basing all our collaborations with biomedical engineering. My name is Roberto Gedaly, Chief of Transplant at the University of Kentucky. In the United States, there are now more than 120,000 patients on the waiting list for kidney transplantation. And only 17,000 kidney transplantations are performed in this country each year.
Immunosuppression is vital in organ transplants, but the drugs that dampen the immune system enough for a successful transplant also have significant side effects. Gedaly wants to shift the role of immunosuppression from drugs to the patient's own cells.
What we would like to do is we would like to create a novel form of immune suppression using patients' own cells and get them reinfused in order to minimize the side effects from the compounds that are currently used for immune suppression, or eventually stop the immune suppression altogether, hoping that the patient will keep these organs for much longer. There's only 5 other protocols like this in the United States. This is probably the first in the state of Kentucky. I'm aware of only 12 other centers in the world using this technology to try to induce tolerance.
This clinical trial will enroll 12 potential kidney transplant recipients. Gedaly's team will use the patient's own cells and special methodology to increase the number of regulatory T cells.
So basically, in this clinical trial, what we do is we send the patients with end-stage renal disease before the transplant for leukapheresis to extract white blood cells. And then what we do is we isolate these high CD25 Treg cells. And in order to isolate these cells, we need to use special beads that have iron. And we have discovered, this is we already have filed for a provisional patent on this invention, that they can actually work as intracellular contrast. In the trial, what we do is we expand the cells. We can go from 40 million to up to 1 to 2 billion Treg cells and infuse the patient with a much higher dose. This is actually real world data. This is one of the first patient that we have been able to infuse. This is actually patients' Treg population pre-infusion. And after we infuse the Tregs, we actually are seeing this new population of cells that when we actually do the phenotyping, they're high in CD25 and FOXP3, which means we're actually seeing a peak in the circulation of regulatory T cells that actually can increase the number of regulatory T cells in the allograft, which could induce tolerance. This is a new protocol. This is a first-in-class study. We're under discussion with the NIH for a UO1 grant application. The novel thing about this trial, we're only going to do isolation and direct infusion into the allograft, which has never been done before. And the reason I'm bringing this up is because we're going to have to infuse directly into the portal vein, and we will probably need a pump where we can actually infuse at a steady pace rate and also monitor pressures. So everything comes from these iron beads that we use to isolate these FOXP3 cells that are high in CD25. They are iron conjugated beads, and they get internalized, as you actually saw. And that's the actual collaboration that I have with Dr. Chapelin and Kurana from Biomedical Engineering and Abdominal Radiology. This is what she showed. The more you increase the cells, the more you're going to see an increased signal on T2 weighted images on MR. And also you can see that the iron gets internalized into the cells and can be used as intracellular contrast. This is my last slide. This is the new protocol that we're actually writing as a UO1 grant application. We'll probably need a pump which actually will have to provide steady flow and probably be able to monitor intraportal pressure. I'd like to acknowledge my collaborators, and I'm open for questions.
Well, Dr. Gedaly, thank you for an interesting presentation. I mean, and congratulations on the work. I know that you have, you and your team have big plans for this technology, this innovation, and are will be very competitive for additional funding sources. So congratulations.